Welcome to the very first A-level biology video on the channel, and we're basing this video on the Golgi apparatus. Now, the Golgi apparatus is an organelle found in a cell, looks like a Spotify Premium / Wi-Fi symbol, if it rings a bell, and it's over here. The Golgi apparatus could be likened to this very niche reference. Now, this niche reference is to GTA 5. If any of the viewers know about Los Santos's custom, where you go into the car, you drive your car into the workshop and you can have different parts added to the car, different car parts, look, the appearance, everything can change. Now you can draw a similar notion to this the cellular structure called Golgi apparatus. Now, before we explore the Golgi apparatus, we need to appreciate the rough endoplasmic reticulum, and the rough endoplasmic reticulum, the rough ER or the RER, has ribosomes located on it. And these ribosomes help with protein synthesis. Well, they're the location of protein synthesis. But what the RER does helps fold protein. So let's say this is our protein, a folded protein. So you see the RER folds protein. That's one thing to appreciate. Now these proteins get carried in something called a vesicle. So the vesicles are almost like carriers, just bubbles to which contents could be carried. Now you have vesicles in a lot of contexts, such as synapses. Just before the neurotransmitters are released, there's vesicles that fuse with the membrane and discharge to allow for neurotransmission. So that's one example. But in the context of within a cell, we've got the RER, it folds the proteins and they get carried. These proteins get carried by vesicles and get delivered to the Golgi apparatus. Now, let's annotate on the Golgi apparatus. We've got the cis face over here, and underneath we've got the trans face. And these inner foldings are called cisternae. The cisternae, because of these sacs in decreasing size, as it goes from top, well, increasing size as it goes from top to bottom, the increased folding allows for increased surface area, and so on and so forth. Now let's talk about the main objective of the Golgi apparatus. The main objective essentially is to modify proteins, and number two, package proteins. Now, the modifications achieved by aggregating or adding carbohydrates, add some carbs. So let's say if you add a carbohydrate, the protein out on this end, on the transphase, the sort of output of the Golgi apparatus would be a glycoprotein. Not only could you modify the protein with the carbohydrate, but you could add a lipid or cholesterol. So these attachments help modify the protein. Now the vesicle part is in relation to the packaging of protein. So we have located over here, circled over here, vesicles, and those vesicles would contain the modified protein. So vesicles are the carriers. So if you were to summarize the goal of the Golgi apparatus, it would be two words: modification and packaging, but you'd exclusively link it to proteins. Now, these vesicles, once they come out of the transphase, they will traverse or go to the membrane to which this process occurs, where the vesicle would fuse with the membrane, and in doing so, discharge all its contents. So this protein. Does anyone know what this process is, where we'd leave the cell? So think about your prefix there. Would it be endo, would it be exo? And it's a process in which you pinch off the membrane and release the material. Yes, you're right, exocytosis. So whenever we talk about vesicles and how vesicles fuse with membranes, it's important to pinpoint the event of exocytosis. So let's take it from the very beginning again. We've got the rough endoplasmic reticulum, you've got folded proteins, these get carried by vesicles. The vesicles then go to the cis phase of the Golgi apparatus, you've got cystinae, and in between those sections, those sacs, you've got modification occurring, where you add carbohydrates, add lipids, add cholesterol, add different biological molecules to that protein. Then once you've got your product, the product will be packaged by these Golgi vesicles represented in this red box here, a red circle, sorry, and then we can talk about exocytosis later on. So this would summarize the activity of the Golgi apparatus, and equally, not to mention the rough endoplasmic reticulum at the beginning, and at the end, the exocytosis event.